Good morning, everyone. So in this today's session, we'll discuss about chapter 17, that is inheritance. This topic is important in terms of your board examination also. Cambridge generally ask 2 to 3 multiple choice question from here, minimum 2 to 3, then 10, minimum 10, maximum 20 mark question from this section in paper 4. And in paper 6 they can ask a general uh, cross related to the genetic disease. So let's see what we have to study in this topic in the current syllabus of IGCSE biology, paper codes 0610. So first, this chapter is chapter number 17, and we have subtopics here. So we'll talk about chromosomes, genes, proteins, right? So in which we'll see how the genetic information travels. What is gene? Then gene is a length of DNA. Define allele as an alternate form of the gene. Describe the inheritance of sex in human. Then state the sequence of base in the gene which determines the sequence of amino acid and that is used for protein synthesis. Explain that different sequence of amino acids give different shape to the protein molecule. That is very important. Then we'll see how DNA controls cell function. That means cells makes all the protein inside the body, including your enzymes, your membrane carrier protein, your receptors, and other neurotransmitters. Then we'll see how protein is formed. Limited to these points, the genes coded for protein remains in the nucleus. Messenger RNA is copy of a gene. mRNA molecules are made in the nucleus, moved to the cytoplasm. The mRNA passes through ribosome and the protein's molecules are assembles, then a specific sequence of amino acid is formed, which is going to form protein in the cell. After that, we'll talk about explain that most body cells of organism contain same gene, but why they behave differently. So we'll discuss about that in this section. Describe a haploid cell limited to the nucleus of the cells and the chromosome number. Describe diploid cell. Then state that diploid cells, there is a pair of each chromosomes, right? Like 23 pairs, total 46 chromosomes. Then we'll discuss about mitosis 17.2. So in mitosis, we'll talk about the nuclear division. Then we'll talk about the role of mitosis in the body in the sense of growth, then repair of the damaged tissue, replacement of the cells, and asexual reproduction. Then we'll talk about the replication of the chromosome before mitosis and why it is important. And then we'll talk about the copies of chromosome get separated, maintaining the same chromosome number, and stem cells are unspecialized cells that divide by mitosis to produce daughter cells. In meiosis, we'll just discuss about how the gametes are formed, and this is a type of reduction division because the chromosome number is half in the daughter cells and they are genetically different also. Then we'll come back to the Mendel inheritance, that's a monohybrid cross. We'll discuss about the genotype, phenotype, homozygous, then heterozygous. Then we'll talk about dominant allele, recessive allele. Then we'll talk about how to interpret pedigree diagram, then genetic diagram to predict the results based on these two 1:1 ratio and 3:1 ratio. You'd Punnett squad in cross, which results more than one gamete to work out, and explain how to use test cross to identify unknown genotype. Then we'll talk about the codominance, example, blood group inheritance, sex-linked inheritance, right? Then in sex-linked, we'll talk about colorblind and use genetic diagram to predict the results of monohybrid cross involving codominance and sex-linked disease. So this is pretty much about the syllabus what we are going to discuss. So without wasting time, let's begin with the topic. Okay. So here's the topic. So let's continue with the topic. So agenda for this topic is, first we'll talk about what is inheritance. We'll talk about chromosomes, genes, and proteins. Cell division, we'll talk about mitosis and meiosis. And then we'll talk about monohybrid cross. Fine, so let's begin with the topic. So when we say inheritance, see, inheritance is a way how the genetic information is going to pass on from one generation to another generation, right? Which leads to continuity of the species and also brings variation in the generation. So it's quite very important that whatever information we have, we can pass exactly in the same order to the next generation, right? Because whatever we do, wherever we go, whatever we eat, right, it's not going to disturb your genetic material. So genetic material is type of memory, you can say. They have the deep rooted memory, right? They do only those for what they are made. Like take an example of human chromosome or the human DNA will work for human. Cat DNA is different from human DNA. Of course, some common proteins are going to form, but that is different. So wherever you go, right, if you're going in any part of the world, right, your genetic material, your genetic information is not going to change. Of course, it is going to just pass on. Whatever you eat, whether you are vegetarian or non-vegetarian or whatever you eat, that is not going to change your genetic information. So it's a very unique thing. It's confined to the generation, confined to the organism. Now the question is in which form these genetic information are present inside the cell. So of course, in the presence of in a chromosome form. So chromosomes are a long, you can see here in this diagram, it's a long, thick filamentous structure, right? And it's a rod-like shape where all the DNA content is being stored. It's like a store thing. So chromosome is like a vehicle, right? And DNA are the passengers, and the driving force is the gene, right? Gene controls everything. So gene is the short length of the DNA, right? Which is found on a chromosome that code for a specific protein in the cell, such as collagen, enzymes, hormones, antibodies, right? So all these things are formed by the gene itself. Out of 100% of the human DNA, 99% are the junk DNA, we call them, because they are not coded for any specific protein, but just the 1% of the DNA or the gene responsible to bring this much manifestation in an organism, right? So yes, genes are very powerful. So now the question is how genes, the long thread of DNA being stored here in a very small gap in a very small container called nucleus. So in every eukaryotic cell, you will find a nucleus present inside. And when the cell is going to divide, the DNA present in the nucleus get condensed, right? So they will get condensed to form chromosome. So this is chromosome, right? So chromosome can be represented like this. This is called karyotype, right? Karyotyping is a technique in which we present the chromosome on the basis of their shape and size, right? So there are Total 23 pair of chromosomes are present here in this colorful diagram, right? So you can see the chromosome through electron microscope, but can also be seen in the cell when they are dividing, right? So normally when the cell is not dividing, at that time, these genetic information are not, they are present in a thread-like structure, like a thread, right? But when the cell is going to divide, at that time, the DNA get condensed in a long filamentous structure called chromosomes, right? And in human body, we have 46 chromosomes. And in sex cells, means reproductive cells, and gamete cells, they have 23 chromosomes only. Now, if you see the male karyotype, see this left one and the right one, male and the female karyotype. So in male karyotype, you can see there is X chromosome and the Y chromosome. Right? You can see some of the chromosomes are red in color, some are blue. So what they're trying to say that one set of chromosome is coming from mother side, another set of chromosome is coming from father side. Both are present in a pair form, right? And we call these pair form as a homologous chromosome. And in the human body, there is 23 pairs of chromosomes in which one set is coming from mother side, another set is coming from father side, right? So what is the possibility of getting boy or a girl in the next generation? So if you'll do the cross, right, the male produces two type of gamete. They have X and Y chromosomes. Maybe gamete contain X chromosome, maybe gamete contain Y chromosome, right? And there is X, one type of gamete produced by the mother, by the female. So if you can cross, you will get 50% chance of getting a boy and a 50% chance of getting a girl in the next generation. So this is how we cannot blame anyone, right? So male is responsible for having a male child, because who will give Y chromosome to the next generation? Of course, the male is going to give. So male is responsible for defining the gender in the next generation. Now let's talk about DNA and gene. So genes are the length of the DNA that codes for a protein, and these genes are present on DNA, right? So see, the information is going like this. So this is the gene, and they will make a protein. And the process by which they make protein is called translation. So this process is called translation. Right? So translation is very important here, right? Next, let's talk about the gene flow. So you can see here, first the DNA is going to make a copy of the RNA molecule. So this RNA molecule, which is produced here, is non-genetic RNA. When we say non-genetic, so generally we have three types. So number one, we have mRNA called messenger RNA. Number two, we have tRNA, transfer RNA, and number three, we have rRNA called ribosomal RNA molecule. And all these three things will help to get protein in the cell by the process of translation. Right, so information is going to travel like this. DNA will make RNA, RNA will make protein. Right? DNA to DNA formation is called replication. DNA to RNA molecule called transcription. RNA to protein forms called translation. Now let's talk about the protein synthesis inside the cell, how the protein synthesis takes place. So first of all, if we have to prepare a protein, let's see the DNA. DNA is present inside the nucleus, and they can't always come out of the nucleus and go in the cytoplasm to instruct the cell organelles what they have to do. So the information always flow in some secret codes, right? And of course, DNA is the mastermind, so they are not going to go out of the nucleus to give the function to the cells, right? So DNA cannot travel out of the nucleus to the ribosome. Ribosome is a cell organ and they performs protein synthesis. So protein synthesis is done by ribosome. That is why we call them as a protein factory. So the gene information is transcribed into an RNA molecule, and this RNA molecule is mRNA. And by which process? Transcription. So first, the DNA is going to make mRNA in the cell. That's the first point. Second point, mRNA, now the mRNA have the information about the protein which is going to form inside the cell. So now the question is, can mRNA make a protein within the nucleus? Answer is no. So they must come out. So they will move out of the nucleus and attach to the ribosome, right? Now the ribosome is going to read the information on the mRNA, and that information is given in the form of codons. Codons are the three nitrogenous base present on mRNA. Fine. Each triplet or each codon is actually coded for a specific amino acid. So when ribosome translate the sequence of codons, actually they are translating amino acid by the process translation. Once a long chain of amino acid is formed, you can see here in this diagram, the green chain has been formed, right? This protein will get customized and they will come to the final structure. And in this way, the DNA controls the function of each and every part of the cell, right? So this is the patent question Cambridge can ask in your exams in 2024 and 2025. How protein synthesis perform inside the cell, or define or describe the process. So always try to write in 5 points. First is the genetic information of the DNA is transcribed on mRNA. mRNA move out of the nucleus and goes to the ribosome, where ribosome reads the information on the mRNA, which is mentioned in the three base letter called codon, right? And the sequence of the codon is going to decide the sequence of an amino acid in the cell, and the process by which it is formed is called translation. And at the end, protein will get reach to its final structure, and then they will operate inside the cell, or they will become more functional. So let's see here. So this is this line. Let me use a cursor here. Yeah, laser point. So see, this is the nucleus. This is the nucleus. Inside the nucleus, there is a DNA. DNA unwinds. Gene is copied on mRNA. Now this mRNA is coming out, right? And this mRNA is going to ribosome, where all the amino acids is now combined to form a protein molecule. But actually inside what is happening, the mRNA molecules have a three-letter called codons. You can see here. These codons are complementary to the tRNA. So if you remember, I've discussed there's three types of RNA molecule, right? Whereas mRNA is the important message coming from DNA to RNA molecule. So in one line, you can say that mRNA have the information about the protein which is going to form inside the cell in the form of codon. Who will bring the amino acid during the process of translation? tRNA. That's why the name is transfer RNA, and they are continuously bringing amino acid. And rRNA makes ribosome. So DNA is making everything. DNA is controlling what type of protein is going to form, who will prepare the protein, who will bring the raw material. So everything is being customized and processed in such a way that the process will go in the sync, means in a flow. Haploid and the diploid nucleus. So as the name suggests, haploid. Haploid means half number of chromosomes. And when we say half number of chromosomes, the meaning is the chromosomes are unpaired. Unpaired means they are not in the paired form. They are one set. All gametes have half chromosome number. Generally, we denote it by small n, also called basic set. Example, all gamete cells. You can write egg cells, sperm cells, pollen grain, ova. Diploid cells means a cell have a complete set of chromosomes. That's a number 46 chromosomes are present. And when the cell get diploid, when the two gametes, male and the female gamete, fertilize and zygote is formed, right? So sexual reproduction maintains the same number of chromosome in the cell, right? And we denote diploid cell as a 2n. So except gamete cell, whatever cells present in your body, all belongs to diploid cell. Now let's talk about the mitosis. So mitosis is a type of nuclear division, right? Where you will get two identical daughter cells, 100% same, there is no difference, right? And this process takes place in any cell. So when we say any cell, the meaning is either it's a DNA or, sorry, whether it's a plant cell or animal cell, right? Now the question is how this process takes place. So just before mitosis, first of all, the chromosome in the nucleus is copied itself exactly, form X-shaped chromosomes. That means you can say replication process has been done. Chromosome line up along the center of the cell where the cell fibers pulls them apart. You can see here in this diagram. And the cell divide into two. And each new cell have the same copy of chromosomes. Why it is important? So all cells in the body are produced by mitosis, right? So mitosis is important for replacing all the unhealthy cells, right? Bad cells, damaged cells, where it takes place for growth, for repair, and asexual mode of reproduction. Meiosis, a type of nuclear division where you will get genetically different organism, not like mitosis. And the number of chromosome will be half here. And this halves occurs during meiosis, and so it is described as a reductional division, right? Why it is important? Because it helps in production of the gametes, increase genetic variation of those species, and produce variations for making a new combination of maternal and paternal chromosomes every time a gamete is formed, meaning that when gametes fuse randomly at the fertilization, each offspring have a different gene set and all look differently. No one can say that 100% genetically identical to the old cell. Question can we ask in the division, in the difference? So you can say mitosis where the two daughter cells are produced, which are genetically identical. Here four daughter cells are produced. All are different. Daughters are diploid. Here daughters are haploid. Daughter cells are genetically similar. Here genetically different. One cell division occurs. Here two times cell is going to divide. That's why we are getting four. So in the genetic terminology, right, when we have to understand the genetics of genetic concept, let me introduce some of the basic definition you must know. So let's go one by one. What is chromosome? So chromosome is a thread of DNA made up of genes. Allele is the alternate form of the gene, right? We'll talk about what is alternate form after some time. Then what is gene section of DNA which codes for formation of the specific protein. Haploid nucleus means nucleus containing half chromosome number, right? Example, gamete cells. Diploid nucleus means nucleus containing full sets of chromosome. That means in the 46 form, 2n form. Genotype is the genetic makeup of the organism. Phenotype is the physical appearance of an organism. Homozygous means when both pair of genes are present together like this, right? Either small t or capital T, capital T. Both are homozygous, right? Heterozygous, where alleles are not similar. Dominant, the genes which is always expressed in the generation, written with the capital letter. Recessive, which is not always show the effect, right? And they are written in the small letters. So now the question is how inheritance works, right? So alleles are the contrasting pair of the genes, and we have a two copy of each chromosomes, right? So that means we have two copies of each gene, and therefore uh, two alleles for each gene, right? So one of the allele is inherited from mother, and another one is coming from father, right? So the character which you can see, that's called phenotype, and the character which you can't see, right? That's called genotype. Dominant allele only need to be inherited from any one parent because it's going to express. But if you want that recessive character can come in the generation, of course, both parents should give that particular gene to the next generation. So if there is only one recessive allele, it will remain hidden and the dominant character is going to express. The dominant allele is always written with the capital letter. You always use the capital letter to show them. And small for recessive. So we cannot always tell the genotype of an individual for a particular characteristic just by looking at the phenotype, right? Like we're saying suppose a red flower, right? So red flower means capital R, capital R. That means when both the parents have given dominant gene, then of course plant will be red. But what about heterozygous form? One capital R and then small r. Here, parents have given two different type of genes. This is heterozygous. Okay. So let's talk about the Mendelian genetics, what Mendel has done. The Mendel was first of all born in 1822 and died in 1884. It's nearby 62 years. And actually, he was a monk who used peas in breeding experiment from 1950 to 1960, 10 years. He has invested his life, dedicated his life to the work which he didn't get acknowledged at that time. But yes, now we appreciate his work and all his laws still we follow, right? He published his ideas in 1966, just after Darwin's publish on Origin of Species in 1859. Darwin's new characteristics could be passed, but never knew about the genes or DNA. So DNA and genes were not discovered during that time. The Mendel studied the inheritance of the different characteristics in pea plant, and you can see in the diagram there are 7 different characteristics, right? And all are found in pea plant. So let's talk about the dominant trait. And we know dominant trait is always written with a capital letter. So let's see capital P for purple flower, small p for white flower, capital T for tall plant, small t for dwarf plant, capital Y for yellow seeds, small y for green seed, capital R for round, small r for wrinkle seed, capital G for green pod color, small g for yellow, inflated pod shape written with capital I, constricted with small I. Flower position is axillary, that's dominant, so we can say capital A, and small a for terminal flower. So these are the characters studied by the Mendel, and we'll see what he has done. So when we say monohybrid, right? So monohybrid means when you are studying one character at a time, right? During the process of hybridization. So either you can take any one character, not all, right? Like suppose if you're working on the height of the plant, you must check the height of the plant. Ignore the other character which you can see here. Now, the Punnett square is a useful tool for predicting the genotype and the phenotype of offspring in a genetic cross involving Mendelian traits. So generally we do the cross to show the results. Mendel cross to breed plant that different for a given character. Pollens from true breeding piece with purple flower is placed on stigma of the true breeding plant with white flower. So here they have taken two species. One is purple flower, another is white flower. Might be you are thinking, well, how Mendel come to know that it's a pure plant? There is no other character. By doing repetitive cross for many generations until, like, suppose you have taken two plants and you started crossing. Then for generation you got there is a dwarf plant. Now you don't want this dwarf plant. You just want tall plant. So just start crossing two tall plants randomly. And then a situation will come in life where the entire plant is tall, right? So the F1 seeds were all purple. So when you will cross a purple flower, right? So you'll get purple flower in the first generation. And when you will cross it, then there's a chance of getting 3:1 ratio in the second generation, right? We can also check the height of the plant. Like in this case, you can see then the true breeders are crossed. In F1 generation, all will be tall. But if you're taking, so now this is not normal genes. Capital T small t is heterozygous. So when in second cross, if suppose both gametes are again going to fuse, then there is a chance that 75% will be tall and 25% will be tall. So we will coming back to the own concept now. Sometimes we do test cross and the whole idea of doing test cross to find something. So first of all, heterozygous plant will be tall with the genotype capital T small t. And the short plant is showing the recessive phenotype written as small t small t. And the results will be as under you can see. In this cross, 1:1 ratio is of tall and short. And how they have done? By doing test cross. What is test cross? Crossing heterozygous plant with shorter plant or crossing heterozygous with recessive plants, right? How to construct Punnett square you can see and you can just follow the steps. It will be easy. Identifying unknown genotype. That's the purpose of test cross. So let's take in condition here that tall and short plant is going to cross. And for short, of course, we know is both are small t small t. Now we don't know the genotype of this particular plant, right? So we have done the crossing and we are getting all tall plant, right? But in the second case, when we are crossing, we are getting 50% tall and 50% short plant. And here 100% of the plant were tall. So the question, so the answer is there's 50% chance of the offspring being tall and the rest 50% being short. Family pedigree, to understand family pedigree, you have to understand the symbols. So you can see this is number represents the total number of people in that generation. And the square box shows male and this is female. The white color shows that they are unaffected, right? And they are normal. Horizontal lines shows that it's a mate between these two organisms. So after doing the Mendel's monohybrid cross, he come to know that when a dominant character is there in the generation, always dominant character is going to express. But the law of dominance is not being followed every time. There is an example of 4 o'clock plant, also called Mirabilis jalapa, which follow codominance, right? So when we are mixing purple flower with the white flower, at them we get all pink flower. So now the question is from where this pink is coming? What about the dominant character? So answer is in case of codominance, right? A pair of alleles are not completely dominant or completely recessive. Instead, you will find a new character in the generation. And you can also say that both have an equal effect on the phenotype. And in case of codominance, the notation for the genotype is written like this: CWCW, white flower, both are small. CWCR, pink flower. CRCR is a red flower. Blood group is a second example of codominance, in which AB blood group is the example of codominance. So generally IA and IB are the two dominant genes. When both are present together, both are 100% expressed in the generation. So there are three alleles of a gene which is going to control the blood group inheritance, right? So first is IA, IB, and IO. O represents allele. So IA results in the production of A antigen, IB responsible for producing B antigen, and IO responsible for not producing any antigen on the cell. Right? So when a couple with blood group A and the parents with blood group B can produce offspring with blood group A, so they're asking, is it possible? So of course it's possible only when both are heterozygous. So you can see here, mother is IA, IO, because to make a O blood group, we need IO, IO in the generation, right? And we know that IU gene is given by the mother and the father side, homologous chromosome. So of course, both parents are heterozygous for the gene. Fine, let's solve this. So a couple who has a blood group A and B, right, have 4 children. Each blood have 4 group. Use the symbols to draw the genetic diagram to show the possibility and use the alleles these. So here allele symbols are given. So always use those symbols which is given in the question. So they're saying couple have A and B blood group and the child have all 4 type of blood group, right? So first of all, they are not in the homozygous form. So they are like this heterozygous form. Gamete. So this man will produce 2 type of gamete. IA, IO. This will produce IB, IO, right? So now you can do a cross, make a Punnett square. Right? So IA, IO, IB, IO. So this is IA, IB. This means AB blood group. IA, IO, that means A blood group. IB, IO, that means B blood group. IA, IB, both are going to express, so this will be AB. So you can say genotype is AB blood group, A blood group, B blood group, right? And O blood group, right? Genotype you can write like this. Always use the notation IA, IB, IA, IA, or IO, sorry. So like this, you can solve it. This is the mark scheme of the question. Now let's have another question. A gene for AB blood group allele is this one. A person with O blood group has a parent who have blood group A and B, right? So it's the same type of questions, but here they have changed in terms of saying that O blood group is the children of a parent who have A and B blood group. So you can write this, right? This is simple. Sex-linked disease. Sex-linked, as the name suggests, sex-linked disease. The meaning is the characteristics is present on the sex chromosomes, either X or Y chromosome. So in this topic, we'll just talk about X-linked disease, right? Right. Let's talk about examples. Color blindness is the example of the sex-linked disease in which XC is written as a normal gene. Sorry, it's not this one. You can say only X chromosome, right? You can say X only X chromosome. The defective gene is written as like this. So XCY, that means color blind male. XC normal but carrier. right? So just take this one. XC, XC again is the normal. So let's see, there is no C here, right? Just normal notation, both X chromosomes. Okay, so let's do the question. Some people inherited blood group color blindness and cannot identify certain color. Even in bright light, the genes responsible of the color vision is located on X chromosomes. So here in the question, they have given the notation. So we'll use the same. So for normal is IXB, colorblind, X small b. So people that are heterozygous for colorblind are called carriers. So what is the genotype of the heterozygous female carrier? So it will be X, C, and X. So this is normal gene, this is defective gene. There is no gene for color vision in the male sex chromosomes. State the genotype of colorblind male. So of course, colorblind male will be like this. Now, figure 4.2 shows a pedigree diagram of a color blindness. These are the keys given here. So the round circle, unshaded round circle is a normal vision. Partially shaded carrier, fully shaded colorblind, square unshaded normal male, half shaded carrier male, complete shaded blind male. So person 13 in this particular question, you have to find out what is the genotype, right? So let's see. So here circle means female, female is normal here, right? So let me write X, XX. Boys had defective, so let's see X, C, and Y. Now you can see in the next generation, all child, male childs are normal, right? 6, 7 are normal. That means male means what? X chromosome is coming from mother side, right? And X is normal. That's why they are normal, right? So let's see, X, Y is normal, but here she is a carrier, not carrier, complete defective. So 13 will be, so this will produce XC. 13 is the is a male parent, right? Is a male. So this will be XCY, that means colorblind male. Colorblind is a sex-linked characteristics. Explain why female 4-5 are carrier. Of course, 4-5 because the father has given X chromosome here, right? And mother given the normal X chromosome. That is why the 4 and 5 daughters, I mean the siblings are normal but carrier. So this is the answer. Thank you, and we'll meet you soon with the new topic, and we'll try to bring a session on the question bank, and in this way we will be able to practice for the exams. Thank you.